All right, welcome you guys. We are going to talk about biotechnology today. So I think this is a really exciting field that maybe a lot of you might be interested in if you want to go down, um, careers in biology. This is really where a lot of research is happening and has been happening for quite a long time. So first we're going to talk about kind of what is biotechnology, is it science fiction? It may seem like science fiction, but now it's kind of coming into reality because we're really going to talk about cloning. So I know a lot of people have heard a bit about cloning, but what is it, right? What's molecular cloning versus reproductive cloning? Cloning whole animals versus just, genes or parts of DNA. And genetic engineering, right? So genetic engineering is a huge part of life right now, especially in the agriculture and medicine world, medical world. So lots of stuff happening in the genetic engineering world. And then we're going to talk a little bit about, why is biotech important and how do we use it in medicine and agriculture. And then at the end, we're going to talk a little bit about genomics and proteomics. So just kind of what the differences are between those two things. So what is biotechnology, right? So it's the artificial manipulation of genetic material, and it's usually the genetic material of living organisms, right? And then we use that manipulation to create new compounds or to perform new functions from that organism. So where is this important? Well, we've been able to improve a lot of livestock and crops. So a lot of agriculture, we've used biotechnology, as well as in the medical world, right? So improving our way we make medications and drugs, and we'll talk about a few examples, but also a lot of industrial applications as well. So not only agriculture and medical, but also some industrial applications too. So what are some ways that we can manipulate genetic material and molecules? So one very important thing is that how do we actually get the DNA out of the cells, right? So we actually have to be able to extract that DNA to use it and manipulate it, right? So first you have to break down the cell. So you have to put like a detergent in there with the cell to disrupt that plasma membrane to actually kind of bust it open. And then you want to destroy all the unwanted molecules in the cell, all that other cellular material, so that you can isolate that DNA and then you can extract that DNA. So I have all the steps down here in the picture, and you don't need to know all of the specific steps. Just know that we are able to extract DNA from cells. And essentially you want to break down the cells, destroy all those unwanted molecules. And then it's really kind of interesting that DNA kind of turns into this viscous strand, and you can actually twist it and spool it kind of like spaghetti onto a glass rod there. So very interesting how we can isolate and extract DNA from cells. So another way we can kind of manipulate molecules and genetic material is using a technique called gel electrophoresis. And you guys may do this in lab, and it's a very common, very useful tool in the lab. And essentially what you do is you can separate molecules based on size and charge. So by charge, we're saying positively or negatively charged molecules. Now, essentially, nucleic acids are negatively charged ions. So our nucleic acids are the DNA and RNA, right? And so that's how we can separate it out. And so what happens is you put it, you put the substance in these little wells in the gel up at the top. So at the top of the image, I know this is kind of a dark image, but where those bright two lines are, that's where the wells are. So you're going to insert the material there, or the molecules there, and then you're going to send an electrical charge or current through the gel, essentially. And what happens is the smaller fragments can move further through the gel. And you actually use a dye, so you dye the molecular substance so you can actually see the molecules as they separate out. So the ones that are closer to the well or closer to the top are the larger fragments or the larger molecules, and then the ones that are closer to the bottom are the smaller fragments because they are smaller so they can move further through the gel. So again, you don't need to know, all the real details of gel electrophoresis. Just know that it's a great technique for separating molecules based on size, really, and charge as well. So another really cool technique for manipulating DNA or genetic material is called polymerase chain reaction, or PCR. So this is used essentially to make a lot of copies of just one region of DNA. So you use a special DNA polymerase. So it's very similar to how we do DNA replication. So the process is very similar, where you have to denature or separate that double-stranded DNA so that you have two single strands, and you use primers essentially and that special DNA polymerase to make copies of that region of the DNA. So then you get these multiple copies, so many copies. You can really amplify the DNA. And so why is this important? Well, this is what is really used when we have a very small amount of DNA and you want to analyze it. So such as a crime scene analysis where you have, just a drop of blood, or, a hair strand or something like that, where you need to do a lot of testing on it, but you only have a small amount of DNA. Well, if you make a bunch of copies, then you're able to use all those copies in all the analysis. So we also use it for paternity testing, as well as ancient DNA. So if you find, some ancient organism or something that was a living organism, you're able to do some DNA tests on it, as well as sequencing DNA. Okay, so just some very cool applications for the polymerase chain reaction, and it's used all the time in biotechnology. So now let's talk a little bit about cloning and genetic engineering. So cloning is essentially creating a perfect replica, right? So whether that's a perfect replica of just a DNA fragment in molecular cloning, or are you replicating an entire organism? So molecular cloning was done well before we actually started cloning whole organisms. So essentially, what is molecular cloning? Well, you stick a foreign DNA fragment into a bacteria. And how do you do that is you use this thing called a vector. And plasmids are essentially a very common vector that we use. And a vector is just kind of a term for a way we get something into something else. So you stick that DNA fragment into the vector, so into the plasmid, and then you get what's called recombinant DNA. So then you're able to put that recombinant DNA into a bacteria. Okay, now why do we want to stick it into a bacteria? So for example, this is how we produce a lot of our insulin in the medical world these days, is we actually stick the human insulin gene that makes insulin, so that's a DNA fragment, into a plasmid, which is a vector, and then we stick that recombinant DNA with that gene for insulin into a bacteria. And then we stick that bacteria into a culture, and essentially the bacteria starts producing insulin for us. So it does it at a very rapid rate because they don't have that much DNA. They essentially are little insulin producing factories. And then we're able to harvest that insulin from the bacteria, and it's a way to amplify or get a lot of insulin in a very easy and relatively short amount of time. So we use molecular cloning in a lot of genetic engineering. So I have an example for us on our next slide besides insulin on how we do molecular cloning. So here's another example of that molecular cloning. So again, this is kind of a step-by-step showing you how we do molecular cloning. And I don't want you to, get too caught up on all the details, okay? But essentially what we do is we use an enzyme that essentially cuts the DNA and cuts the vector at a very specific location. And so that means they create this kind of complementary, what we call sticky ends. But essentially the sticky ends just match to each other, right? So essentially you're able to stick that foreign DNA into that plasmid vector because it has a perfect spot for it, right? And so that's how we essentially put that foreign DNA into that plasmid. Now you might say, okay, how do we find that foreign DNA again, right? We've stuck it into the plasmid, we're gonna stick it into a bacteria. Now, how do we know which bacteria actually has our recombinant DNA that we're interested in harvesting, right? So that's what this image is showing, is that essentially we have a gene that's in that plasmid. For example, this LAC-Z gene is essentially a gene for an enzyme. Just call it LAC-Z, right? So we have this enzyme gene that essentially we cut, so that gene no longer functions and no longer produces that enzyme in our recombinant DNA ones, because we've stuck that foreign piece of DNA in there where that gene for that enzyme used to be. So then, once we stick all those plasmids with our recombinant DNA into the bacteria, some of the bacteria may take it up, some may not, right? So how do you decide which ones have our plasmid with our recombinant DNA? And that's how we use then, we use antibiotics as well to separate. So in our plasmid, we have a little ampicillin resistant gene. So essentially, that bacteria, if it's taken up our plasmid, is resistant to ampicillin. So if we plate all of those colonies onto, onto a plate that has ampicillin, which is an antibiotic, it's going to kill off all the bacteria that didn't take up our plasmid, right? Because they're not going to be resistant. And then how do we then tell which one has maybe the plasmid but not the one with the recombinant DNA? And that's this blue and white colonies, and that's where that LAC-Z gene came into play. So essentially you put a substance in it, in that plate with the ampicillin, and essentially it's supposed to change color if it's exposed to that enzyme that the LAC-Z gene produces. So the blue ones are not going to have our recombinant DNA, but the white ones are. So we know that all those white colonies are going to have the plasmid and have our recombinant DNA in that plasmid. So I know that was kind of a complex process, but it's kind of really interesting to think about how you can insert foreign DNA into bacteria. Now, reproductive cloning is very cool. So I know maybe a lot of you guys have heard of Dolly the sheep. She was the first genetically cloned multicellular organism. So we call reproductive cloning making that identical copy of a multicellular organism. And how did we do that? How do we actually do that? Well, you essentially have a recipient animal, right? And in this case, we're talking about sheep. So you take a recipient sheep that's essentially going to grow the clone, okay? And then you have a donor sheep, and that's the one you're cloning. So you're cloning the donor sheep, okay? So what happens is you actually take an egg cell, right, from the recipient, so from that sheep, and you remove that haploid nucleus, because remember we said gametes, or eggs and sperm, are haploid cells, right? So they need to pair up with another gamete to produce a diploid cell. So essentially you trick it, okay? So you take out that haploid nucleus and you implant a diploid nucleus from your donor cell, from any body cell from the donor, which is who you're cloning. Okay, so you essentially trick that egg cell into saying, hey, you've already found another gamete, you now have a diploid nucleus, and you're going to start cellular division. Okay, so essentially you then stimulate cellular division, and that recipient sheep is going to grow an exact replica of the donor sheep. Okay, and that's how we got Dolly the sheep, because she was a cloned sheep. So it's very cool and very interesting and kind of maybe scary that we can actually clone multicellular organisms. Okay, so I know that this is kind of weird, but it's been around for a while, and you can actually clone, if you have an animal a pet or something like that you want to clone, it is possible, which is kind of crazy to think about, right? So now that we know about cloning and kind of the differences between molecular cloning and reproductive cloning, we really use molecular cloning a lot in genetic engineering. That's the most common method on how we manipulate living organisms. Okay, so what do? We actually use that recombinant DNA technology, right? So we have our plasmid with our foreign DNA molecule in there, and then we're going to add it to another organism to modify its own DNA for more desirable traits or other traits, right? So we've seen things that maybe we don't necessarily need to do, but we have and can do. And that's where kind of the moral aspect of genetic engineering takes place. So we actually add that foreign DNA, right, or whatever gene, that desirable trait that we want, we add that using the vector method from molecular cloning into the new organism, and therefore they will express those traits of the new gene we've put in, or multiple genes, right? And this is how we get genetically modified organisms, or GMOs. So I know a lot of you guys have probably come across GMOs in probably the supermarket, right? You go to the store and there's a lot of things that say non-GMO or no GMO, right? Because a lot of people are very worried about genetically modified organisms, especially eating genetically modified organisms. Now, everybody has their own opinions about it and whether it's safe or not, but I have included some interesting articles for you guys about genetically modified organisms and, is it safe, is it not safe? And so everyone can come up with their own opinions about it. So genetically modified organisms, you're putting foreign DNA into an organism. Now that foreign DNA can either come from the same species or from a different species. So if you get it from a different species, that's the now considered a transgenic organism. So it's still a genetically modified organism, but it has foreign DNA from a different species. And I have a really cool video for you guys about the top like 7 or 10 crazy transgenic animals. So obviously, the bottom left pictures are all, fake, right? They're all photoshopped, but they're not that far from the truth, which is the crazy part, because the image on the right-hand side is an actual truly transgenic organism. So they have these rats that grow human ears on their back. So very, very strange and kind of very sci-fi, right? Science fiction, but not so much. So if you watch the video, it's kind of cool just for your own fun edification on what kind of transgenic animals are out there and what we have actually produced. And why? Why make these transgenic animals, right? Is there some sort of medical reason for it? Environmental reason, or is it just because we can, right? So that's kind of the question. So there's this thing called reverse genetics that is very important in the biotech world and genetic engineering, because we don't know exactly what all of our genes do, right? So we're mapping out a lot of DNA in different species and everything, but essentially reverse genetics tries to figure out what genes do by deleting or mutating them. And then you see what happens to the phenotype, right? So in forward genetics, you have the phenotype, right? You know what the phenotype is, and by cloning, mapping, all of those things, you're able to discover the underlying genotype. But in reverse genetics, you know the genotype, but you don't know maybe necessarily what it does or what the phenotype is. And so that's the forward and reverse genetics. So just kind of something to think about, that you can play, with the genome forwards and backwards. So how do we use this biotechnology in medicine and agriculture? Those are probably the two biggest fields that we have seen a lot of advancement using biotechnology. So medical purposes, right? We can study genetic disorders. We can also discover methods to treat diseases, right? Just like we kind of discussed about insulin, how to make insulin for treating diabetes. We use it to produce vaccines, right? DNA vaccines, recombinant DNA. So lots of medical uses for biotechnology. But also we see it a lot in agriculture as well. So we can create GMO plants that are resistant to diseases, resistant to pests and environmental stresses. So that way you can improve on the crop yield and the quality of the crops as well. So lots of advancements in agriculture, and a lot of our plants and fruits and everything that we do eat today are GMO organisms. So just as an example, here on the right-hand side, biotech saved the papaya crops. So they essentially developed a virus-resistant papaya, and this virus was killing all the papaya. So, that you see a lot in agriculture, whether it's a virus, a disease, or a pest, or something like that that is just devastating the crops, then you're able to create a GMO version of that crop, and then you can improve the crop yield and the quality as well. The quality part is maybe the controversial part, right? Maybe does it not taste as good? Everybody has different opinions, right? that tomato that lasts on the shelf for a really long time? Is it actually a higher quality than a non-GMO tomato, for example? So let's go through some of these specific examples in biotech, in medicine, and then we'll do some in agriculture as well. So we kind of saw some broad strokes of how it's important in medicine and agriculture, but let's look at some specifics. So we can actually do genetic diagnosing. So you actually can diagnose genetic defects through genetic testing before you even administer treatment. So it's a way to focus your treatment if you know that it is a genetic defect. So for an example, in breast cancer, anyone that develops breast cancer, or even before developing breast cancer, you can test for this BRCA or BRCA gene, which is an inherited gene that increases your chance of developing breast cancer. So it's an inherited gene. So if you have breast cancer in your family, you're able to actually test for that gene before you may have ever developed breast cancer. And so this little table here, graph, just shows you the risk of breast cancer in different age groups and showing general population versus people with that BRCA mutation. So obviously you have a much higher risk of breast cancer or developing breast cancer if you have that gene. So a lot of people, if they test for it, they know they have the gene, can actually do treatment before they even have any sort of symptoms of the disorder. So it's a way of prevention more than anything else. So another very exciting technique that if we can figure out exactly how to use it and how to make it work, it could be very exciting, and it has the potential to cure a lot of genetic diseases and possibly even cancer. So I have a great video on gene therapy that kind of shows you what it is and what it could potentially do. So what is gene therapy? Well, essentially, it's very similar to how we do molecular cloning, but instead of introducing just a foreign gene, you're essentially introducing a non-mutated gene into the genome of an affected individual with a genetic mutation. So you essentially replace that mutated gene with a non-mutated gene. So then you can replace that protein or whatever that gene that is mutated, you can replace it with a non-mutated gene that's going to produce that protein that may be absent in whatever that disorder is. And so essentially you do this in a very similar method as we discussed in molecular cloning. You use a vector, right? And in this case, we commonly use an adenovirus. So instead of a plasmid, we're using a virus. And so you put that non-mutated gene into the vector, and then you essentially replace that gene in the genome of the individual. So therefore, that instead of making some sort of defective protein causing the genetic disease, you can then create a normal protein and hopefully cure that genetic disease. So again, this is very exciting because it's very advanced and could really end a lot of genetic diseases, especially the ones that are caused by one gene mutations. So again, check out the video. It's very cool. So this is just an animation of how we're using that gene therapy, using that adenovirus as a vector to essentially insert that non-mutated gene into the nucleus or into the genome of the individual cell. So again, you don't need to know all the details, but just kind of a cool image. So just some more ways we can use biotech in medicine are for vaccines. So we've already discussed a little bit about vaccines, but essentially we're able to mass produce such as the flu vaccine using molecular cloning. So you can create a lot more of the vaccine for mass production and mass dispersion of the vaccine, which is very helpful, and especially nowadays when we're talking about trying to develop a new vaccine for this COVID-19. So, how do we, one, get a vaccine that works, and two, how do we mass produce it so that everyone has access to it? So, again, vaccine, I'm sure they're using biotechnology to create a COVID-19 vaccine. Also antibiotics. So antibiotics, we actually produce a lot of them by using a fungi. So what are fungi, right? Like mushrooms. The mushrooms are the ones that are actually producing the antibiotic, and they are so good at it, they actually, improve the yield of antibiotic production. So very interesting ways of how we can improve yield on production of these medications. So now let's talk a little bit more about biotech in the agriculture world. So long before we even had biotechnology, farmers have been really selecting plants with desirable traits, right? So they say, okay, these are the ones that survived that frost, or these are the ones that survived that viral attack, right? So they were already selectively engineering their plants without doing this whole biotechnology and bioengineering. So we've been doing this in agriculture for long before the modern day biotechnology. So genetically modified plants, we can make them disease resistant, right? We can make them resistant to herbicides and pesticides, right? So essentially you're killing off the things you want to kill off instead of the plants. You can even have them have better nutritional values and better shelf lives, okay? So there's a lot of ways we can manipulate our plants for the betterment, for the improvement of the yield of the crop. So transgenic plants, right, receive DNA from other species even, okay? So again, we said we had genetically modified plants, just like we had transgenic animals receiving DNA from other animals. We do have transgenic plants as well. But the thing is they have to really undergo a lot of extensive testing to ensure the ecological stability, meaning is it going to overrun maybe the native species or something like that. So transgenic plants, I mean, obviously transgenic animals too. There's a lot of things that come into play when we're talking about transgenic species, okay? So a big question nowadays, and that we're all faced with every day we go to the store, maybe for not for some people, but maybe for others, yes, this is a huge question, right? Are GMO products safe for human consumption, right? Now, everyone can have their own opinion, and you can argue probably both sides, right? But I just linked an article here, and it's also on Canvas, but the overall finding essentially stated that there's no differences between genetically engineered and non-genetically engineered foods to implicate a higher risk to human health from genetically engineered food than from their non-genetically engineered counterparts. So again, is GMO safe or not? According to a lot of the scientists out there, they are safe, okay? But the big thing is there are a lot of movements out there though that are all for non-GMO, and it could be for other reasons besides whether it's safe or not, right? So lots of ideas out there, lots of opinions out there. But there's also the whole organic idea as well. So what does organic then mean too? So organics have to be are more having to do with pesticides and the products that are used in the growing of the plants. But it does have to do a little bit with GMO too. So if you notice all these different comparisons of organic labels, right? So there's varying degrees of 100% organic to organic ingredients. And one of the big things is the GMOs. So all three of the other ones say no GMO versus the organic ingredients may contain GMO. Okay, so lots of different things about organic and GMOs. So we've been talking a lot about genetic engineering and introducing specific genes here and there to living organisms to kind of, produce desirable traits or things like that. But what about genomics and proteomics? So genomics is the study of the entire genomes. So essentially, instead of just manipulating, one gene here and there or a couple of genes, you're studying complete sets of genes. And you're essentially sequencing the DNA, right? So you're looking at the nucleotide sequence and the organization of the nucleotides. So you're also studying a lot of interactions within a species and with other species. So how do you compare, what similar genes do, cats and dogs have for example, right? So if you're studying the entire genome, you can start sequencing and mapping the genome as well. And that's what we'll talk about next. So in genomics, you can actually start genome mapping, and that's finding the exact location of these different genes on the chromosome. So this is maybe where you're doing forward and reverse genetics because you may find a gene, but you don't know what it does, or you have a phenotype, but you don't know where the gene is. So essentially you're looking for all the genes on the chromosomes and what exactly they do. So this is where you get genetic maps and physical maps, and you can then help that to identify different diseases, right? So if there's certain genes causing diseases, and a lot of diseases are caused by multiple genes. So it's not as simple as maybe it sounds, and that's why we haven't been able to maybe cure cancer or heart disease and say, hey, it's this one gene. With breast cancer, with that BRCA gene, we've definitely come a long way, and we say, hey, if you have this mutated gene, you have a really high risk of developing this disease. But it doesn't mean that if you don't have it, you're not going to develop the disease either. So again, we've come a long way, but we've got a long way to go as well. But we can also identify microbes that have beneficial traits as well as plants, right? So we can produce higher crop yields and have plants that are better at adapting into different environments as well. So a lot of cool stuff happening in genomics and genetic mapping. So when we talk about genome mapping and genome sequencing, the whole term is whole genome sequencing. So it's the process of determining the DNA sequence of an entire genome. Okay, so essentially it's a very problem-solving type approach to genetic diseases. So you want to know the entire genome sequence so you can determine where all those genes are located and maybe what genes are causing these genetic diseases. And so what we actually do is we use model organisms, so simpler organisms and species that are used to study certain things in their genes and then apply them to other species. So an interesting thing that they have found is that fruit flies um, are actually behaviorally act the same way as humans do when exposed to alcohol. So they actually process alcohol the same way as we do. And so there's a whole study that was done on fruit flies and alcohol processing and the behavior in fruit flies. So I actually linked the paper to it because it's kind of interesting. But we use the mouse a lot, right, as a model organism. So essentially we just use other species to study, and then we can then apply what we find in those model organisms to other species such as humans. So how do we apply genomics, right? So we've been talking about genome sequencing, whole genome sequencing, genetic mapping, and so on. And so essentially we have what's called the Human Genome Project, and there are other groups that have expanded the applicability of DNA sequence information. So how do we use this human genome information for predicting maybe disease risk, right, at the individual level? So maybe we know a disease risk at the population level, but what about individual level? So is it an increase because of the genetics, an increased risk because of the genetics, or is it environmental? So this is how we can kind of debate the two. You know, if you find a gene that's responsible for it, then maybe the genetics are causing an increased risk, or is it environmental? Okay. So there's a lot of these genome-wide association studies where they're actually looking for these associations between genes and disease to try to provide some data for other studies. So, you know, some diseases we have found associations with genes, but a lot of them we haven't. So there are these kind of, you know, big studies trying to look for more and more genes that are associated with diseases. And that's part of this Human Genome Project. So we can also use genomics to create new biofuels. So if we look at the genomics of microorganisms, we can find a better way to harness biofuels from algae and cyanobacteria. So biofuels are a renewable source of energy. So if you are able to strain the algae, and which then converts the carbon into this energy-rich fat, you're able to process all of that into biodiesel. So it's a way to kind of, you know, how do we make this algae and cyanobacteria work for us better, right? How can we make it more efficient and create some new options for biofuels? So it's kind of interesting. I have a video here that I'll also post in Canvas. So proteins, as we have found out, are the final products of genes, right? So after we've undergone transcription and translation, we've translated the genetic information into the building of a protein, right? And so these proteins are going to play very important roles in our cells. So when you talk about a proteome, okay, so we have proteins and then we have a proteome. And a proteome is the entire set of proteins produced by a cell type, right? So we have all the same genetic material in all of our cells, but the proteins are going to be different because remember we talked about gene expression and the control of the gene expression in our cells. Some cells are going to produce certain proteins while other cells are going to produce other proteins. So some genes are turned off in some cells and turned on in other cells. It just depends on what the purpose of that cell is in that specific tissue. So proteomics is the study of the function of those proteomes. So if a proteome is all the proteins produced by a cell type. You're gonna study that proteome, and that's proteomics. Okay. So proteomics studies the function of those proteins produced by a certain cell type, right? So this has come into play for cancer research and cancer therapy. So we can use it to improve screening, early detection for cancer with biomarkers and protein signatures, which are just groups of biomarkers. So essentially you can say, okay, if this type of cancer cell we know is actually going to produce these types of proteins, we can look for those proteins. Okay, so I know that's kind of complex, but essentially we know now, studying proteomics, right, and studying the different proteomes of different cell types, we know, okay, a neuron, right, is going to produce this proteome. They're going to produce these proteins, right? And that's normal function of a neuron cell. Now we have a cancerous neuron cell. It doesn't produce the same proteins. It may produce a certain protein that we can actually look for and screen for in early detection. So we call it a biomarker, right? So you're looking for that protein that isn't normally produced, okay? So I know that that's kind of a complex idea, but it has come a long way, and it's definitely helping with cancer research and therapy. So speaking of proteomics and cancer research and cancer therapy, we've actually discovered that animals can actually detect early cancer and other disease, maybe even better than we can. So essentially, dogs are able to smell, at a certain level that humans obviously are just not capable of doing. But they are able to detect maybe that protein or that biomarker that cancer cell is producing very early on in the disease, okay, which is very, very cool. So I have a really cool video about these dogs that are able to actually smell and identify these proteins that are being produced by these different diseases. So check it out. It's very cool. Um, and there's a whole little story about a lady that was saved by her dog who could detect essentially early cancer in her. So let's finish up with a little of self-review so you guys can just kind of go back through the slides and answer these questions. So what is genetic engineering, right? So what did we define it as? And how has it been used in the medical field and for agriculture? And then what is the difference between genomics and proteomics, right? I know they sound very similar. So what are those key differences? So you guys can go ahead and answer those on your own, and go ahead and check out the videos that are posted on Canvas as well. They're very cool. They're very exciting. They're not going to be on any exams, but they're just really fun. And this is a very exciting field in biology that I think you guys might be interested in. So, all right, until next time, I'll see you later.